So it's a pleasure to introduce Anindya as a speaker here today for departmental seminar. And I guess this is Anindya's third lecture or second lecture as invited.
Third, I think.
Third lecture. And as has already been probably clear from our discussion, that Anindya is a person of high sophistication. And he's a fantastic teacher. And he's a very able researcher as well, which is statistics that I would present would definitely tell. Anindya obtained his PhD degree from IICS Kolkata with Professor Konkon Bhattacharya as his thesis advisor, and his PhD was done in 1998. And then he joined as an assistant professor in IIT Bombay in 2002 and promoted to full professor in 2012. So far, Anindya has published about 130 papers in international peer-reviewed front-ranking journals, plus 2 book chapters. And 17 students so far has got PhD under his supervision, and currently 9 students are enrolled. And as I said, that Anindya is a nice teacher, and that's why he has got recognition in teaching, Department Award Excellence in Teaching in IIT Bombay, and some other professional accolades he has got. And his involvement in the science and physical chemistry has been recognized by international community as well. He is in the editorial advisory board of the Journal of Physical Chemistry for 2 years, 2020 to 2022. And he is a member of board of studies of various reputed IITs and institutions, including a couple of IITs. With this, I would invite Anindya to deliver his talk on excited state phenomena in fluorogenic molecules and semiconductor nanocrystals. Anindya, please.
Thank you very much, Ranjit, for the kind introduction. And thanks to Rajiv, Ranjit, Shumon, Manik, and others who have been kind enough to invite me to speak to this August gathering. This is the third time I'll be speaking in SMBO Center, but I've visited many times. There's so many friends there. And I mean, all of the research that gets done there. So it is really a pleasure and an honor. I'll try and keep my talk within 40-45 minutes, no more than that, and then we can have questions. But looks like some students are there. Please feel free to stop me and ask questions if you want. What you see here is more or less the present group. 2 of them have actually graduated and gone for their next destinations. But this was the group in March. We're standing in front of Powai Lake. We might be having an alligator. We'll have more to talk about that at the end, lurking somewhere in this grass. So we live in a veritable jungle. And behind us, we can see another kind of jungle, a concrete jungle, which is Hiranandani. I thought I had turned it off. Sorry about that. Now I think we have taken care of it. So that is where the rich and famous stay. So I like to say that our students graduate from our natural jungle and go and stay in the other jungle many times. I'll talk about a couple of problems that have kept us busy over the last maybe 6-7 years or so. So what we are looking for in what I'm going to talk about, a large variety of chemical systems, is we are looking for systems that are going to give us light, that are going to be light emitters, but they're going to be light emitters when we want them to be, in response to certain physical or chemical inputs that we provide to them. And to this effect, we studied 2 kinds of systems. One is fluorogenic molecules, molecules that are not so fluorescent by themselves in solution, but they can become fluorescent upon complexation with a metal ion, or upon formation of aggregates, or taking it a little further by formation of crystals, or making a thin film out of them. Application of such fluorogenic molecules is diverse. They can range from sensing of analytes to making organic LED kind of devices. we are not really into applications. We are trying to understand the phenomena, proper manipulation of which may lead to an application someday in future. The other kind of system that we have been somewhat reluctant entrants to are photoluminescent semiconductor nanocrystals. Quantum dots are things that we do not have to introduce anymore. We now have things like quantum dot television sets and all. So everybody knows that when we take some semiconductor material and start breaking it up into smaller pieces, then upon going down below a limit, we see what is called quantum confinements, and we see discrete energy levels appearing in the conduction band as well as the valence band. And when that happens, life becomes very colorful as this photograph right here. I think all of us would know that color of these quantum dots depends on the size. It can be described more or less to a great extent by the simple model of particle in a box, where the particle is exciton, the box you can think is the nanoparticle diameter. And here also we are trying to understand certain things. What kind of exciton dynamics take place? What are the time constants? How one can manipulate them to get light out of it when we want it. So that, in summary, is going to be the crux of the talk that we'll have over the next 45 minutes or so. Now, to do this, we use mainly time-resolved spectroscopic techniques. Once again, to this audience, I suspect we do not really need to introduce these. If you want to look at fluorescence or photoluminescence dynamics in nanosecond to hundreds of picosecond time scale, then we use this highly celebrated method called time correlated single photon counting, TCSPC. So, and if you want to go down to hundreds of femtoseconds to a few picosecond, maybe 100 picosecond or so, then we use what is called femtosecond optical gating. Both these instruments are actually there in your institute, but just to make sure that everybody is on the same page, the crux of the matter in both the cases is we excite whatever it is that we want to look at by using pulsed light. So the light is on for a very small time, typically 100 femtosecond or so, 100 or 200 femtosecond, and then it goes off. So what this pulse does is that it creates an excited state population all of a sudden. And then when the light is gone, this excited state population is left to decay on its own. And in the simplest case scenario, the decay is of first order. This is the same integrated rate law for first order reactions that we have studied. It's just that instead of population, we have written fluorescence intensity I at time t and I at time 0. Time 0 is the time at which the laser pulse hits the system. And the other difference is that conventionally, we don't write the rate constant k, rather we write the time constant tau. And in case the decay is single exponential, this time constant tau is the average life spent by the system in the excited state. So it is called the lifetime. Lifetime, once again, for single exponential decays, is actually a measure of how brightly fluorescent or how brightly photoluminescent our sample is. The relationship between emission quantum yield phi f and the lifetime tau is simple. The proportionality constant is the radiative rate constant. Similarly, if we write 1 minus phi f on the left hand side, the constant is going to be KNR, the non-radiative rate constant. The excited state can decay by radiative as well as non-radiative pathways characterized by their own particular rate constants. So this is how, by recording spectra, determining fluorescence of photoluminescence quantum yield and recording lifetimes, we can actually get an idea of the rate constants of the radiative and non-radiative processes that are involved. Professor Devashree Ghosh is here. She has done a lot of work using quantum chemistry on these non-radiative processes of some very interesting class of molecules, as others like Professor Biswas and Rajiv. Now, the thing is, it's not always that we have a single exponential decay. More often than not, the decay deviates from single exponential nature. So the most used and perhaps the most abused uh, fitting model that is used is a multi-exponential model. Multi-exponential model actually works in many cases, but more often than not, uh, this multi-exponential kind of function would fit decays that should actually be something else. Today we will not get into this, but hopefully next time we meet, I'll have more to say about this. The other kind of experiments that we do is transient absorption. Here, once again, we create an excited state population by an intense laser pulse. The laser pulse here is actually more intense. What we see here is this black box here, well, literally black box, is our amplified laser that provides femtosecond pulses at 800 nanometer at 1 kilohertz repetition rate. The white box here is optical parametric amplifier that can give us many different colors, still pulsed. And this here is the transient absorption spectrometer. What happens in the spectrometer is that this intense pulse is focused onto the sample, which is kept somewhere here, and another weaker pulse, which is typically broadband, we call it white light, is also made coincident on the sample. So, and what we do not monitor the pump afterwards. So, this big pulse is the pump that is just stopped after going through the sample. What we monitor is the probe pulse, and we look at delta OD, which, to put it very qualitatively, tells us how this spectrum, absorption spectrum, changes as a result of pumping. What happens is, first of all, you can get a negative signal in the region where there is ground state absorption. This is called ground state bleach. And recovery of this ground state bleach with time tells us how quickly or how slowly we get back the ground state from the excited state. We can get positive signal, which is due to absorption of the excited state. Decay of this transient absorption signal tells us how the excited state population decays. And also we can get another kind of negative signal, which we're not going to discuss today, that is due to stimulated emission. Now, all these things can happen in probe because we are looking at a broad range of probe light, broad range of wavelength. So the actual transient absorption spectrum can look something like this or something that is even more complicated. Analysis of transient absorption data has to be done with great care, and one needs to look at many different wavelengths at the same time, and one needs to think what kind of model would be suited. Finally, for the last couple of years, we have been looking at fluorescence lifetime under a microscope. This instrument here is from PicoQuant. It's called MicroTime. It's essentially a confocal microscope, but the excitation sources shown here are pulsed diode lasers. So one can record lifetimes like this from a very small volume. One can use these lifetimes to generate nice pseudo-color images like this. What you see here is lifetime images of pollen grain. Additionally, one can do what is called fluorescence correlation spectroscopy. You don't really need pulse lasers for fluorescence correlation spectroscopy, but it helps to have it because along with FCS, you can do many other experiments if you have pulse lasers. We'll present only small amounts of data from this instrument today. When we meet next, perhaps it will be exclusively on this. And this is perhaps the last introduction slide before I start talking about our own work. In excited states, molecules often have very different reactivity from the ground state. So phenomena that don't happen in ground state take place in excited state because the electron configuration is different, if I put it very, very simply. So after excitation, let us say there is a proton transfer or there is a charge transfer, there is photoisomerization. One of these processes can take the molecule from the locally excited electronic state to what I like to call a nascent state. Let us say both locally excited state and the nascent states are fluorescent. The nascent state must have energy that would be lower than the locally excited state. So lambda 2, the characteristic emission wavelength of the nascent state, would typically be longer than lambda 1, the characteristic emission wavelength of the locally excited state. So how do we know that this has happened? So first of all, the excited state phenomenon would provide an additional non-radiative depopulation of Le, and it would lead to population of Ns. And this leaves its signature in a steady state as well as time resolved data. In steady state, if this here is the situation, in absence of the excited state process, we'd say we'd get a single emission band. In presence of the excited state process that takes the molecule from Le to Ns state, we'd get the longer wavelength emission as well. Uh, if you look at it in time domain, if you look at time evolution of fluorescence intensities, then if you monitor the fluorescence decays in the blue end of the spectrum, so here we get a major signature from a locally excited state, and that now decays quickly because of the additional deactivation channel that is proposed. We see a fast decay, and the Ns is not even there at the moment of excitation. Population of Ns actually grows for some time from Le, and then of course whatever goes up has to come down. So we get a plot like this, which involves a rise time. The situation here is very similar to the time evolution of the population of an intermediate in the chemical reaction, where we start from a reactant, go to through an intermediate to the product. That is something we have all studied in 11, 12, first year, and so on and so forth. So this is the signature of fast decay and in the blue end, and rise time in the red end. Now, combining the steady state and time resolved data, one can construct the time resolved emission spectrum, which can show us how the emission spectrum changes as a function of time post excitation by pulsed light. Incidentally, these data that I'm showing and that I use for teaching purposes always, the data are quite old, and the first time I ever showed this data, when they were new and actually going to be published, was 2005 January, and the venue was SN Bose Center. So I had actually forgotten, but then while preparing for the talk today, it occurred to me that there is a historical connection between this data and SN Bose Center as well. Now, the first kind, one of the most celebrated kinds of excited state phenomena is intramolecular charge transfer. When we excite, uh, there is a charge transfer, and you can create a dipole moment by pulsed excitation. So at the moment of excitation, the dipole moment is sort of switched on very quickly. My ex-student Avinash, who's now a postdoc in Canada, started this work, continued by Sharmishta, who's about to graduate, and Projit, who's a current graduate student. So we have been looking at these amino quinolines for a long time. Depending on which positional isomer we take, they have different behavior. And 5-aminoquinoline, especially in protic solvents, exhibits a very strong polarity dependence of the non-radiative rate constant. ET30, as many in this audience would know, is a empirical micropolarity parameter. So what happens when we switch on pulsed excitation? And when I say this, I say it a little bit of trepidation, because it is sort of like taking coal to Newcastle. I'm going to talk about solvation dynamics, and I'm going to talk about solvation dynamics to an audience which has Ranjit and Shumon and Rajiv, and in the same institute we have Shomesh. So they have done phenomenal work in this field. So what happens when we switch this dipole moment on? Remember, this is a solute. We have very dilute solution of this in some solvent. Let's say it's a polar solvent. When we switch the solute dipole moment on, the solvent dipoles actually respond to this and try and reorganize around the solute dipole. In the process, due to favorable dipole-dipole interaction, we have lowering of energy. Now, if you remember, this dipole moment is there because the molecule has been excited. So energy of the excited state is lowered gradually due to solvation. And it is not just dipole-dipole interaction. In case there is some specific interaction like hydrogen bonding, that would also have a role to play on how fast or how slowly the solvent molecules reorient around the newly created solute dipole to have the best possible energy situation. That is marked by time-dependent Stokes shift of emission spectra. Uh, and the way usually solvation dynamics is studied is to plot C of t. C of t is essentially a measure of how much of solvation is yet to be accomplished at any given time. When we plot that as a function of time, we get a decay, and from there we can extract the time constants associated with solvation. Now, the specific problem that we tried to understand using 5-aminophenolene, since it did prove to be a good solvation probe in neat alcoholic solvents, is we thought we are going to take very small amounts of polar solvents in a non-polar bulk, say 0.1% or 0.2%, 0.3% of methanol in hexane or diethyl ether, something like that. So essentially, the solute of fluorescent probe would be to start with surrounded by nonpolar molecules, and we'd have these polar molecule dipoles scattered over or clustered among themselves or something. So when we excite, we create the solute dipole. And then what happens is that the solvent dipole moments actually try and displace the nonpolar molecules around the newly created dipole and try to form a solvation shell. This phenomenon is called preferential solvation. And using 5-aminophenolene and by a very careful analysis of data that Sharmishta had done, we figured out that first of all, the time associated with preferential solvation is about a nanosecond, and that involves all these different kinds of motion, translational diffusion, formation of the shell, deorientation, and all those things. And we could also point out that specific interactions do have a role to play. Now, we have only done the experiment. To understand this more completely, molecular dynamics simulations are in order. It appears that my friend and Guru Bhai, Shobhan Sen, is interested in doing molecular dynamics of this system. Once they do it, if they do it, perhaps we'll have a clearer picture of what is going on. But as of now, this is where we stand, and we are extremely happy that this work has been selected as our contribution to the virtual special issue of Journal of Physical Chemistry that is being published to honor Professor Kankan Bhattacharya, with whom I had the good fortune of doing PhD. As most in this audience would know, one of Professor Bhattacharya's major contributions is the slow solvation in interfacial regions. And there has been phenomenal work over the last 20-25 years in this direction in so-called biological water. I remember more than 20 years ago, the thrill of hearing from Shamit when he came to Indore for some time. He told me about how they studied this slow solvation in DNA protein adducts. That's absolutely thrilling. And then there's been a lot of work on this kind of slow solvation in biological water and in other systems like ionic liquid and so on and so forth. So what we did, we're actually not worked in this field for a long time because we wanted to do something else. But then Professor Kamendra Sharma, my colleague, is an expert in a very strange kind of system. So what they do is they take a protein, they cationize the surface, and then they put a negatively charged polymer surfactant on top of it. And this polymer surfactant typically is a liquid in itself. So you have a protein polymer composite in such a way that the protein is completely engulfed in the polymer. Of course, it is not as if it is one protein molecule and one polymer molecule. It is a milieu. So it actually resembles a crowded system. As if that was not enough, one can remove the solvent. I mean, one can remove water and then end up with a viscous solvent. I'm going to show you a photograph soon. So this is famously called waterless liquid proteins. It is liquid, but it's claimed to have no water. Water is there, but very small amount. Professor Stephen Mann of Bristol had done pioneering work in this. Kamendra was Professor Mann's postdoc. So when Kamendra joined and we learned about the system, we became curious to know whether if we have this kind of biological water-like behavior in these so-called waterless systems. This work was started by my postdoc at that time, Tara Shankar. Anushwa is Kamendra's student. So to start with, we wanted to investigate this by taking a probe, fluorescent probe, that is non-covalently bound to the proteins. And we did not really get any signature of solvation because there was a particular region in which this probe would always go. But then, since an expert like Kamendra was on board, we did make some interesting observations. But then we thought, can we see slow dynamics if we use covalently labeled protein? So after many years of thinking and some false starts, my student Tanuja actually has observed that kind of signature. So as you see here, Tanuja has observed slow decay of C of t over a few nanoseconds also. And we see different curves here because these are at different stages. What happens with the protein? Then what happens with the cationic protein? That is number 2. What happens when we put the polymer on top? And what happens when we remove water completely? We do see slow dynamics at different extents in these different systems. And Tanuja has also performed FCS studies. And this year, a couple of papers were published, one in theory and one in another kind of experiment, which seem to be in agreement of what we think is going on here. What we think is going on here is maybe it may be a little too premature to talk too much about it. Let me just say that we do get slow solvation, but we do get a fast component as well. We are yet to understand the system completely, but it looks like a very interesting system to study. That being said, let us move on to the system of fluorogenic molecules. My ex-student Tuhin, who is now a postdoc in Berlin, started this field of work in our lab. We started working with this shift base. Now, what happens with this shift base is that they have very flexible structure. So the fluorescence quantum field is always very small, and if you can somehow block the movement, the associated non-radiative processes would be cut out. And that is when you can hope to get strong fluorescence. So that was the motivation of studying molecules like this. And looking at this structure, the first thing that would strike any chemist is that this is a good chelating agent. In fact, it's used by inorganic chemists to make inorganic complexes. So the first question we asked is that, is it possible to make a complex with a metal ion which is strongly fluorescent, more fluorescent than the free base ligand? And the answer was yes and no. For only with zinc and aluminum, we got brightly fluorescent complexes. Secondly, if you look at the quantum yields, the quantum yield is about 10 to the power minus 4 for the free base ligand. There's an order of magnitude increase upon complexation with zinc, almost 2 orders of magnitude increase from there upon complexation with aluminum. So complexation with any other metal gives non-fluorescent complexes. So why is it that the aluminum complex is more strongly fluorescent than the zinc complex? That is the question we had, and the answer seemed to be that the aluminum complex is a monomer. The zinc complex is a dimer and a pi stack dimer. So the two sulfur moieties come very close to each other. So a lot of things can happen between them. You can have X-dimer formation. You can have a host of other processes. We do not really know why, what exactly are the processes that happen there. But the experimental result we have is that when it is dimeric, it is not as fluorescent as it is when it is monomeric. But then we wanted to test this hypothesis. So then what we did is we made crystals wherever possible, or we made thin films in solid, and we tried to see what happens. What happens is interesting. Cellophane, which has a picosecond lifetime in solution, has a nanosecond lifetime in its crystalline form. The aluminum complex, which has a nanosecond lifetime in solution, has a picosecond lifetime in crystalline form. So, and that is because when you look at the crystal structure, aluminum complexes, well, upon complexation with aluminum, cellophane becomes planarized, and this is how the planarized complexes stack in the crystals. So this seems to be a circumstantial evidence for our hypothesis that in this kind of complexes or this kind of molecules, Pi stacking kills fluorescence to a large extent. So then we thought, can we introduce small functionalities like methoxy group or ethoxy group and disrupt this nice packing in case of the aluminum complex, and then do they become strongly fluorescent? The answer was yes. We introduced these methoxy and ethoxy groups. When we looked at the crystal structure, they are no longer nicely stacked, they are no longer planar, and the result is we get strongly fluorescent solid with nanosecond lifetime. So then we were left with this question: is it possible that zinc in these cases always forms dimeric complex of some kind or the other? So then we thought, is it possible to make zinc complexes that are strongly fluorescent? So the challenge was to find zinc complexes that are not pi stacked. And that is where my student Shorodip came in, and this work is being carried forward by Orko Prabha. Shorodip found out from literature that Salampy, another Schiff base, was reported to form zinc complexes like this, or the report was monomer, but this is what we got, depending on which anion one used. So both the zinc complexes are such that in one unit you have two Salampy molecules, fine, but they are not stacked. They are either side by side or they are perpendicular to each other. And the result is that we get significantly stronger, longer lived excited states for the zinc complexes. It perhaps shows up better in nanosecond time scale. This is the fluorescence decay of the zinc complex. Both the zinc complexes have the same lifetime. This here is the fluorescence decay of the aluminum complex. The lifetime is 6 nanosecond. With cellophane, the lifetime of zinc complex was like tens of picosecond, and now we have 6 nanosecond. So difference between 6 and 9 is significantly less than the difference between tens of picosecond and 1 nanosecond that we had between zinc complex and aluminum complex of the earlier Schiff base. So looks like if we avoid pi stacking, we do get longer lifetimes. But why is it that the aluminum complex still has a longer lifetime? We did literature survey, and what we learned was that apparently with aluminum complex one gets more rigid. Apparently with aluminum one gets more rigid complexes for Schiff bases. And here the idea is that more rigid the molecule, more rigid the complex, more fluorescent it is going to be. So that is what we think it is as far as the complexes are concerned. But on the way, we found out that even before we make the complex, if we just deprotonate the Schiff base, Salampy, we get a significantly longer lifetime. So if we now again come back to the nanosecond time regime, this here is the decay, fluorescence decay of the zinc complex. This is the fluorescence decay of the anion. And you cannot even see the fluorescence decay of the free base Salampy molecule here, because it practically gets over in 50 picoseconds. So the anion itself is significantly stronger emissive. If you look at the quantum yields, 0.003 for Salampy, 0.04 order of magnitude increase again for the anion, one more order of magnitude increase upon formation of the complexes. And the lifetime associated, lifetime of this excited state, fluorescent excited state of the anion is about 800 picosecond. Upon performing experiments in faster time scale, we saw signatures of solvation and most likely conformation and relaxation. So it appears that for the anion, solvation and conformation and relaxation prepares the emissive excited state, which has a characteristic lifetime of 800 picosecond. So we have built for this class of complexes a roadmap of how to go from fluorogens to stronger fluorophores, depending on the kind of input we provide. And this stronger emission in case of anions is not just restricted to Salampy. We tried it out with cellophane and another Schiff base, which is called HBAP. In both cases, we observed the same kind of phenomenon. I'll not talk about the low temperature data because already I've spoken for 31 minutes. We are doing a lot of experiments in this area, but let me now go to the next class of fluorogenic molecules. We were lured into working with this class of molecules, long name, let's just call it DBMPT. We were lured into working on this class of molecules by my colleague, Professor Anil Kumar. The work was started by my then postdoc Anusha, who is now a faculty member in Odisha, another ex-postdoc Shomodipto, who's now a postdoc in the lab of Professor Dohal, and Sharmishta. You'll see Sharmishta's photograph in almost everything. Yes, she has done a little bit of everything in our lab. Now, DBMPT, once again, uh, it has a very flexible structure, and associated with all this motion are plenty of non-radiative processes. So, if you can quench them, if you can stop that motion, you can hope to get fluorescence. And that's what we see here in acetone solution. Acetone is a good solvent for this molecule. There is no fluorescence. If you add little bit of water, then first of all, the mixture becomes turbid, and we get significantly stronger fluorescence. We got sort of nanorods from this system when you added water. But the problem with this solvent mixture experiment, as we found, was that it was very difficult to quantitatively reproduce conditions. So we tried to clean up the system a little bit, no pun intended, by adding surfactants. So when we add surfactant to DBMPT in stoichiometric amounts, we are not talking about an experiment where we have 1 micromolar of the probe and millimolar surfactant. We are talking about a situation where they are present in more or less same quantities. Then we see that we get this nice, good looking nanorods, which one can see not only under electron microscope but also under confocal microscope. And we were a little worried that perhaps these nanorods come out only when you remove the solution, but then we saw them in cryo-TEM as well. Using fluorescence lifetime imaging microscopy, we could follow the morphological evolution of these nanorods, and we could see the modal lifetime going from something like 1 nanosecond to something like 3 nanosecond. This is very preliminary data. We are trying to understand this in more detail, and some kind of simulation would be required to understand why we get nanorods, and that's all the more so because of the results we got with triblock copolymers. We used two kinds of triblock copolymers, and we got two kinds of structures. With P123, which has a smaller hydrophilic uh, chain length, we get microspheres, and with F127, which has longer hydrophilic chains, we get nanorods. And again, we followed the morphological evolution, and we found that the nanorods can penetrate cell membrane while the microspheres cannot. Uh, Once again, we have no idea why it is that in one case we have nanorods, in another case we have microspheres. It is got nothing to do with the uh, triblock copolymers themselves, because the triblock copolymers, in absence of DBMPT, both of them form small, relatively smaller micelles that are spherical in shape. So the shape and the size, that they come because of interaction of DBMPT with these triblock copolymers. We have no idea why they are different. We are talking to some chemical engineers who do simulation. Uh, I don't know whether we'll be able to figure out that way the reason for the different morphologies. So far we have talked about enhancement of fluorescence by arresting segmental motion of the molecules. Another highly celebrated way is to use plasmonic nanoparticles. So we'd like to use that uh, technique as well, but right now we are just learning how to go about them. We started this work in collaboration with Professor Alison Funston of Monash University. Bala was a very good graduate student in a joint graduate student between IIT Bombay and Monash University. Hemen is carrying this work forward. Now, for the next maybe 10 minutes or so, let me talk about our efforts to understand this very exciting field of semiconductor nanocrystals. I mentioned that we were sort of reluctant entrants. We were reluctant because by the time we entered this field, so much of work had been done. But we still decided to study this because just from our idea of photochemistry or spectroscopy of organic molecules, we don't really understand all of this splendid kind of literature that is coming out in this highly interesting field. So we are doing this research not because we are going to make quantum dot solar cells that is going to solve the energy problem of the world, or because we are going to make quantum dot television or something like that. We are just trying to learn uh, how to go about understanding exciton dynamics in this kind of nanocrystals. And so many things can happen. So first of all, when we excite, this much I think this is something that all of us know, we create an exciton where we have an electron in the conduction band, a hole in the valence band, and they're coupled. Now, we typically excite at energies that are beyond band gap. So, you create hot electrons and hot holes, so they can cool down. That time is typically associated with 0.32 picosecond. Then electrons can get trapped. Typically, from literature, we know that that time is can range from 2 to 30 picosecond. Hole can also be trapped. So, there are different defects in the nanocrystal. Crystal has to end somewhere, and that is where we get defects. So, especially at the surface, there are plenty of defects depending on their chemical nature. They can capture electron or they can capture hole. So this hole trapping is again a very fast process. And then we can have shallow trapping, deep trapping, a lot of things. We can have something called Auger emission, uh, Auger recombination, where an electron can just pass its energy to another one while recombining with a hole, and the second electron can be promoted. So we started our work with this cadmium selenide nanotetrapods in collaboration with Professor Rohit Srivastava, our colleague from Bioscience and Bioengineering. Some of you might be familiar with his work. Rohit is one of the recipients of SS Bhatnagar Prize this year. And we started with this very energetic, that time MSc student, Ashutosh. Shucheta's entire thesis is on cadmium selenide nanotetrapods. She's graduated. She is an assistant professor in a postgraduate college in Pune. And Faryad is a graduate student who is taking this forward now. The reason why we chose cadmium selenide nanotetrapods is twofold. First of all, this was like one class of nanocrystals that was not studied using spectroscopy to a very great extent. Now, the structure and all were known. Woodside structure in one place, some other structure in some other place. Why is it that the rods grow? So all these were established. But and it was proposed that it can have many diverse applications. But not much of work was there on time resolved spectroscopy. So again, Bala contributed a lot in this. He had some experience coming from IISER Pune. So we were able to make two kinds of tetrapods. The one in the bottom, the B panel, was something that showed only bandage emission, and the one in the top was dual emissive, showed bandage as well as trap emission. And when we looked at the dynamics, PL dynamics, we found that when we had only bandage emission, the photoluminescence decayed with a time constant about 8 picosecond. There are some minor long components as well, but by and large 8 picosecond. When we have the trap and when we monitor bandage emission, we observe an additional 1 picosecond component, which we assigned to the trapping time. And when we looked at the trap emission, the redshifted emission here, the lifetime associated with this emission was pretty long, tens of nanosecond, and as you can see, it is definitely not exponential. So in this, we also did transient absorption studies, and we got our fingers wet, and we got to understand how to do these experiments and how to make sense of the data. However, the quantum yields associated with these nanotetrapods was abysmally low. Then we thought, is it possible? And here we learned a lot from the work of people like Narayan Pradhan. We thought, is it possible to dope the nanoparticle, knowingly add some ion or something, which would lead to separation of the electron and hole wave function, and does that give strong photoluminescence? This was already reported in spherical nanocrystals. The challenge was doing it in nanotetrapods. So with a little bit of effort, Shucheta and then Faryad was successful in making copper doped nanotetrapods. And we were very happy to note that these tetrapods had a very strong what is called charge transfer CT emission. It is copper 1. It is doped with Cu1, which has been established by Faryad by uh, titration with a whole scavenger as well. So very strong emission, CT emission, associated with a lifetime of hundreds of nanoseconds. Why does that happen? By a careful analysis of transient absorption data in different ranges, uh, this is the scheme that we came up with. In doped nanocrystals, the Cu plus trap, Faryad gets really unhappy if I say trap, so let me use the word he likes. When we add Cu plus, it captures a proton in a matter of 1 picosecond or so. And that is what competes with the relatively slower process of trapping that is there in the tetrapods already. So the reason why it works is that the whole capture, did I say proton? Sorry about that, whole capture. Uh, the whole is captured by copper at a faster rate than the inherent hole traps that are there. And it is then the recombination that takes place eventually, that is what is long lived. So that is what we understand about the doped CDAC nanotetrapods so far. Some other work is in progress. Now, of course, one of the things that we wanted to see is there any application of this in biological systems, especially now that we are doing microscopy. Now, it broke our hearts to note that if we exchange the ligand and make the nanotetrapod water soluble, then it becomes completely non-emissive. There is no photoluminescence at all. We studied its interaction with proteins and we showed that it can displace molecules like warfarin bound to proteins, but to be very honest, it was no fun. While all this was going on and we were sulking over not having photoluminescence from our water soluble nanocrystal, Shomodipto and Sharmishta sort of conspired and they decided to make spherical nanoparticles in water. So this entire synthesis is done in water. Uh, they used polyethylene imine as the capping agent, and they were successful in making cadmium sulfide quantum dots capped by polyethylene imine, and these quantum dots, one can see, are strongly photoluminescent. The photoluminescence spectrum is really broad because the PL is dominated by trap emission, different kinds of traps perhaps, and the lifetime is again hopelessly not exponential, but associated with a long time constant. The very careful and backbreaking analysis of the transient absorption data yielded the time constants associated with all the processes that take place post excitation, and to give credit to Sharmishta for doing all of this. The other thing that we observed is we wanted to know, is it possible to modulate, because remember the theme of our work is light emission at will as per order. So can we quench this emission by specific inputs? And we found that among the metal ions that we studied, only lead can quench the photoluminescence of these cadmium sulfide nanoparticles at concentrations as low as 10 micromolar. Uh, this is an analysis that we did reluctantly because we knew it doesn't work, but then one of our reviewers insisted that we do it, and well, finally we could make a statement out of this. See, whenever we see quenching, the classical approach is to construct Stern-Warmer plots. The issue is that the Stern-Warmer plots are very, very nonlinear. The plots that we get using steady state PL intensity and lifetime are very different, and the quenching is too strong. X-axis here is in micromolar. So if you calculate the bimolecular quenching constant maybe from the initial part or something, we get an absurd high value. So, and we say it's absurd because see, conventionally what we think of dynamic quenching is that it is a diffusion controlled process. Now, of course, a diffusion controlled process cannot really have a rate constant that is as high as this. So we knew that it doesn't work, but well, we ended up demonstrating that Stern-Warmer analysis is not really what you would like to do when you talk about PL quenching of nanoparticles by whatever, the approaches that one can take are, first of all, one can think about what is the correct decay model. As we said at the beginning, multi-exponential decay model is the most used and perhaps the most abused model. So that is one thing that we are thinking, and here we have help from papers published in 1965, 1970. People have actually proposed many different decay models. We are studying them and we are trying to see whether we can go that way. The second thing that one can do is one can use complementary techniques like transient absorption. That is what we did. Once again, by a backbreaking analysis that I'm not going to go into today, we established the mechanism by which the quenching takes place. It involves an ultra-fast hole capture followed by a relatively slower electron transfer as well. And the reason why there is selectivity for lead at such low concentrations is that the PI is a strange customer. It does not like to interact with lead. So lead is able to get through this capping agent and reach the surface. While the other metal ions that we studied, they love PEI. So they get trapped and they are not allowed to approach the surface at all. That is why they cannot work. If they reach the surface, they would have quenched the PL as well. More work is going on in this direction. Professor Leela Srinivas Panchkarla's group has made another kind of CDS nanocrystals capped by olailamine. And here, Hemen has noticed something very interesting, single exponential decays with time constant of 11.6 nanosecond or so at the emission maximum. If we record the PL decays in the blue edge, we do get faster decays. Once again, the emission spectrum is broad and is dominated by trap emission. Now, being fluorescence spectroscopists, seeing data like this, what we have tried to do is this. We have tried to generate the decay associated spectra. Right now we have fitted to 3 exponentials, which I am not completely sold about. I think it is actually a by exponential decay. We are trying to see whether we can do that. The long component, 11.5 nanosecond, that gives us a spectrum that is exactly the same as the emission spectrum, steady state spectrum. However, if you look at the short component, 0.6 nanosecond or so, we get a significantly blue shifted and narrower spectrum. That is because that could be the bandage emission. It's possible that other phenomena are involved here, but it could be bandage emission as well. So this is something that we are trying to probe. Meanwhile, Sharmishta is also not idle. She has now made copper doped cadmium sulfide nanocrystals in water once again. And she's trying to study the dynamics using fluorescence correlation spectroscopy. Again, it is a little too early to talk about this. Next time we meet, it is possible that we'll have more to say about this. To conclude, I'm sorry I've taken 5 minutes more than what I promised. The theme of our work is to try and play around with different excited state processes and to tune light emission as per our requirement. One of the intriguing phenomena that we are trying to follow is the slow solvation in solvent mixtures and waterless liquid proteins. With Schiff bases, we have established that rigidification leads to stronger emission. Pi stacking seems to be a hindrance, and anions definitely emit from a solvated excited state. So we are trying to put all this together and trying to see whether we can now think of proof of concept applications in fluorescence sensing using this kind of molecules. Similarly, with DBMPT, which exhibits what is fashionably called aggregation induced enhancement of emission, we're trying to think whether it is possible to make them sense things like viscosity or polarity or something like that. And with semiconductor nanocrystals, we have learned how to use complementary spectroscopic techniques to get a complete picture of exciton dynamics. Let me put on record my thanks for all the students present and past, postdocs present and past, collaborators at different times. And here I'd like to thank 3 people especially: Shukendu Nath of BRC, Jyotishman Dasgupta, I did not correct their spelling, sorry about that, and my ex-student Shiva in IIT Goa. A large fraction of the audience is experimentalists, and as we know, one of the biggest nightmares of experimentalists is prolonged breakdown of instruments. Right now, we are going through such a phase where our transient absorption is not working because one of the lasers is broken and is being repaired. So these three kind souls are helping us out by letting us work in their lab. And of course, our lab is always open for whoever would like to use whatever little we have there. No work can be done without funds. Let me thank all the funding agencies. Thank you all for your patience. I hope that once this horrible period is behind us, we'll get to meet many of you in our beautiful picturesque campus. Thank you very much.
And we have that promised alligator somewhere there.
Yes. This is an alligator. The picture was taken by my colleague, Professor Balakrishnan.
Thank you, Anindya Datta, for a really nice talk, and I'm sure particularly the students have enjoyed a lot. So now the floor is open for questions. So please feel free to raise your hand if you have a question. Or you can type in the chat box. Yeah. So Professor
Anindya Datta,
yes,
very nice. Yeah, just a kind of clarification that when you talk about the shift base, you say that they are kind of mobile, and that's why their quantum yield is less. So
yes.
What makes the quantum yield less?
So what happens is when you excite anything, any molecule, if they have a flexible structure, then you have non-radiative processes that are associated with that motion that can take place in the molecule. Flip-flop motion, or in this case, photoisomerization also happens. So if we make them rigid, then this motion is stopped, and consequently, these non-radiative pathways are blocked off. And as lifetime is 1 by kr plus knr, knr becomes small. That is why tau becomes large.
Okay, so this is a general. For all the molecules, this can happen. No?
Yes. See that is why pyrene is so very strongly emissive,
right?
Whereas these shape bases are not.
Exactly. Very nice. Thank you, Anindya.
Thank you, Rajiv.
Okay. Maybe while we wait for other questions, I'll just ask my question. So I have a few clarifications I was curious about. So one thing is about these tetrapods. So what is the origin of that that directionality? So from the center.
So what happens is you have the woodside structure in the core, and after the core has grown to a certain level, some of the planes of these woodside structures serve as seeds for the protrusions that take place. So it is energetically more favorable for those protrusions to take place rather than the core growing in size.
Okay.
That is why we get
different surfaces grow at a different rate.
That is correct. That is correct.
Okay. And then the other question is, so when you talked about these quarterless proteins, it's a very curious system. So, but it's a mixture of, I guess, protein with other surfactants or other.
It's a protein with a polymer surfactant.
Okay. So then how do you know that where that water is? So is it really near the protein or it's trapped by some other species? So is there any way to...
So first of all, the bulk water is not even there, because what is done is you make this and then you put it in this freeze-thaw cycle and remove it. So maybe 5 ml of solution becomes half an ml of a thick viscous liquid. So that water is gone. Whatever water is there can only be trapped between the protein and the polymer.
I see.
And the question I have personally, and that is what I have not understood, is that Professor Bakchi had written this very nice commentary a few years ago. I've forgotten the name, but it's a lovely, very well written commentary for the layman, where this question was posed, and of course, you people know this much better, that how much is the role of water and how much is the role of the movement of the protein chain. So what I feel is that in this kind of system, perhaps one can try to understand that question as well, especially because not much water is there.
Sure. Yeah.
And that was really the motivation for starting the work.
Sure. And my last question was, it was again kind of, I'm curious to understand that when you talked about this preferential solvation, you mentioned that it is possible to distinguish between, so there are two roles. One is that long range electrostatic interaction with the dipole-dipole interaction, that can be a long range component, and also there is a short range specific interaction component. So is there any way to dissect between the two other than doing the MD simulation? So
I doubt very much. See, experimentally we get to know that it happens, because if we use acetonitrile as a polar solvent, and when we use methanol as a polar solvent, we get different dynamics. So it does happen, but I think to dissect, as you said, perhaps MD simulation is the only way.
Okay, thank you. So
in fact, that was a major comment we received from the reviewer of that paper, that why are you not doing MD simulation? So finally, we had to throw up our hands and say we are not doing MD simulation because we don't know how to do it.
Sure. Okay, so that's all from me, and I think Devashree has a question. Please.
Yes.
A very nice talk.
Thank you.
I have a question about one earlier part of the talk. So in those stacked configurations, actually I would expect that the stacked configurations would be more rigid, and therefore it will be more fluorescent. How come that's not the case?
All right. First of all, stacked conformations being rigid has been demonstrated in a family of organic solids by Professor T.P. Radhakrishnan's group about 10 years ago, and others have also shown it. In our case, the only reason why it would not be fluorescent is that new non-radiative channels open up involving these two ligands that are in close proximity. It could be excimer formation, non-radiative, non-emissive excimer formation, or it could be something else. We don't really know what is going on there. That is what we are trying to find out. So, but opening up of additional non-radiative channels is the only possibility that can explain this.
Another thing is, in that same line, then how general do you think that stacking would lead to more fluorescence that kind, or less fluorescence that kind?
What we see is that it is general for shift bases, but it is not really general across all family of fluorogenic molecules.
Oh, I see. Okay.
Is there any other question?
Sir, I have a query here. Can I ask, sir?
Yeah, please go ahead.
Yeah, thank you, sir. Sir, it was a very nice talk, sir. Thank you very much, sir. Sir, I have one question from your initial part. While you are showing the dual fluorescence, is it because of two different excited state, or it is because of same excited state but two different conformer?
What I showed was because of two different excited states, but in fact that is the more general. You can think of two different conformers if they are stable in experimental conditions to be associated with two different excited states as well. What I showed was, let us say, proton transfer is my favorite excited state process. So let us say you excite, and then the molecule loses a proton. So then, in the excited state, suppose you are working with a phenol. That's even easier to understand. So in the excited state, by loss of proton, you create a phenolate ion. So for me, the locally excited state is the phenol excited state, and the nascent state is the phenolate excited state.
Okay, sir. Okay, sir. And sir, another question, just I would like to get your suggestion that, like, you showed us both organic molecule as well as inorganic molecules. Now, which one is like most be applicable?
Depends on what your application is. There are many inorganic complexes that have really, really long lifetime. That is why such complexes are attractive for many applications in photodynamic therapy or in lighting devices and other things. But fluorescent molecules, the good thing about fluorescent molecules is that you don't have to worry about things like presence of oxygen. The problem with involving a triplet state usually is that if there's any oxygen, they are going to cause deactivation of the triplet state. You will not see that long lifetime, long-lived species. So it depends very much on what application you are trying to look at.
Okay.
Thank you, sir. Thank you very much. Sir, one comment can I have, sir?
Oh, sorry. Yeah, go ahead.
Okay, sir, Dr. Devashree asked one question that aggregations. Sir, we can have no HH aggregation which will reduce the intensity and JJ aggregation which which can increase the intensity. So I think that that can be the answer.
Yes, it is possible, but what we seem to be having here is that in addition to the expected H or J aggregate effect, we seem to have new non-radiative pathways opening up. This is something that is typical to our class of molecules.
Oh, thank you, sir. Thank you very much, sir. Thank you. Nice talk, sir.
Thank you very much, Pralad.
Yeah, I think Shwikot has a question. Please go ahead.
Yes, sir. Sir, is the rigidification of the sheaf bases to be fluorescent depends on the charge of the metals at which it binds with the sheaf base?
I don't know yet. What? So first of all, the problem is that even if we take anything other than D0 and D10 configuration metals, we don't get any fluorescence enhancement. You only get quenching. That is one thing.
Sir, but because
aluminum is 3 plus and zinc is 2 plus. That's what you're saying, right? Yes. So,
yes. So the charges.
I don't know. I really have not thought about that. What we think, and we are trying to understand that. See, there has been some work which says that since sheaf bases have oxygen as well as nitrogen, they form more rigid complexes with aluminum compared to zinc because of hard and soft acid base kind of interaction. So seeing this, what we thought is that if that is the case, then what happens if you use boron instead of aluminum? You should get something similar. And then we did a little bit of literature survey, and we find that this boron sheaf base adduct, these are actually being studied. The work started in 2017. Professor Tilagar of ISC has done some work in that direction. We have also made some of the complexes. So far, in those that kind of adducts, most of the work that has been done is more of synthetic value. We are trying to study it in a. We are trying to do the photophysical study. So let us see what comes out of that. Once that is done, perhaps we'll be a little closer to being able to answer your question, Shwikot.
Okay, sir. Thank you.
Anindya,
Yeah, Ranjit, please go ahead.
if I don't ask you a question, then Shumon will not talk to me. That's what Shumon told me.
All right, I will also not talk to you.
Yeah. So of course, I will ask you a question on the solvation dynamics part. Okay,
Yes.
that's easy for me. The thing is that you saw a very slow solvation, right? For the case of this surfactant and then protein dressed with positive charge and all those things. So in that solvation part, do you have any dipolar contribution? Is there any part which is coming because of the initial part, the faster part is coming from the dipolar orientation? Is there a possibility?
Perhaps it is there, but the question is, how do we know? Without MD simulation, is it possible to figure it out?
No, just kind of qualitative hunch. If your protein has some kind of dipolar moiety and that is rotating itself.
We think that is very much the case. In fact, so
right.
Ranjit, that is why we started doing that work.
Right.
What we thought is, and also while starting this, another thing that occurred to me was the very first slow solvation paper we published from Confinements lab. That was in zeolite, no water. So in zeolite, we saw very slow
Yeah, I know.
Yes.
Yeah.
Not only sodium plus.
Zeolite.
Exactly. And there, one of the things that we proposed is that it is, first of all, you have these free ions that solvate the fluorescent probe. Also, the backbone cannot move, but the fluorophore itself can move.
Right, the self motion of the solute.
Exactly, that is also. So that is one thing that I had in my mind. That do we have something similar in this very different system?
Yeah.
And my hunch is that yes, we do.
Okay. Because the ionic part is understood, and you know that dynamics in ionic solutions by Mark Marancelli in 1995.
Right.
All those have shown you that slow component arises from the translational dynamics of the ion. So maybe it is.
So here, something similar. I mean, dipolar components of the protein. Most likely, they also have a kind of a slow deorientation, and that can contribute to the slow solvation there.
So you are using the intrinsic fluorescence of the protein to?
No, we are using a CPM level protein.
Okay. So that is covalently linked, right?
That's right.
Okay. So maybe the protein motion itself, but in that case, whether that is contributing to the initial first part or the slow part depends on which part of the protein it is tagged, and
Right.
how slow or fast the motion is compared to the transitional motion of the ion.
To complete this work, what we would like to do is try and label different parts of the protein and then see whether there's a difference in dynamics. Similar to our collaborative work with Samrat published a few years ago, where we had tagged an intrinsically disordered protein in different regions. And we saw different kinds of solvation dynamics there.
But then that will lead to another controversy that not every part of the protein is equally exposed.
Very true. And here, this is a different thing altogether.
Right. So, very difficult. Anyway, thank you.
Okay, if there are no more questions or comments, let us thank Professor Anindya Datta once again for really nice and illuminating lecture. And we hope to welcome him in person under better circumstances, hopefully in near future. So, of course, you have an open invitation to visit us anytime. So, yeah, we look forward to seeing you in person sometime soon.
Same here.
With that, let me thank all of you again for being here till the end of the session.
Thank you.